point.
Yeah. So we'll make a point to tweet for him today. And yeah, let's welcome Brent.
So thanks, Dave and Luke, for inviting me. It's been a great visit. I mean, really a good vibe in this department. I'm very impressed. Lots of good science here. So it's been really, truly wonderful, even with the snow. I love the snow. So rather than talking about, one long thing for from a paper that you will read or won't read, or while they're reading, I thought I'd do 3 small topics, really little tasters, so you can get a feel for what I do and you can follow up on it. So start off with acknowledgments. So my work's very collaborative. Right, so we work with Jeremy Beaulieu, Brian Karstens, Nathan Jackson, Arietta Morales, JJ Chai, Stacy Smith, Pam Diggle, and Charlie Fenster, and funded by various people, for which I'm grateful. Okay. So 3 topics today. One is diversification, what drives floral evolution. One's phylogeography, one's protein evolution. Okay, so what drives floral evolution? This was an 8-year-long collaboration at a nascent working group. This lasted from when I started my PhD, from when I started my postdoc to when I got tenure. It was this long collaboration with this working group, and resulted in this one paper. It's actually the paper I'm most proud about in my career, right? Because it has a good fusion of biology and methods, and actually just people working together from a variety of fields to get at questions. Okay, and so the main question is, why is the world the way we see it? Why are there so many flowering species? What causes different traits to be common on the tree of life? Okay. And of course, this isn't a new question. Ledyard Stebbins asked this and did started looking at various key characters commonly used in plant classification to see if they're clumped, right? And so things like, calyx and corolla both present or absent, right? Petals present, absent, and so forth. Okay. And then a quick little table, right, showing all these character combinations in their frequency, right? So this number here, 12, means 12 families have resumed ancestral state for these all 8 traits, right? And 36 have this combination, and none have this combination, right? And of course, this is not phylogenetics, so all of you should be horrified, right? It's the 50s. It's a crazy time. And so it still sort of shows that, like, if you were just dropping things randomly, it probably wouldn't be this clumped, right? There's something happening here, right? And so his idea was that this clumping is due to combinations of characters from like large families, right, evolved through natural selection, right? So you see these key combinations because they're awesome, right? And selection favors them. Okay. But continuing questions about this, what's the mechanism for this? Is it natural selection? Is it differential diversification, right? Which traits matter most? Right? Does petals matter, or is it something else about where the ovaries are? Is the pattern truly non-random? Right? Because we know stuff's clumped because of phylogenetic history anyway. Right? So this is actually a thing. It's just sort of a random pattern. Okay. So our group got together and settled on 6 traits instead. Right? Corolla present, corolla absent, radial symmetry, and so forth. And then went out and chose 500 species, made a tree for those species, and scored the characters. Right, so always the question of sampling bias and things like that. We had a very complex sampling scheme. It was a big pain in the neck, but it was to make this really as pure as we could get sample. Okay, we got a sample of 500 species, and then we can look at all these combinations. Right, so this is a little table. So here shows corolla present, and so black is the presumed ancestral state, and then green, purple is derived. Right, so we have in this combination with corolla present, perianth unfused. Few stamens. Okay, so you have that distribution, and you can we can count those across our taxa, right? And get this histogram. Okay, and we see a whole lot of these, right? And a few over here. And just pointing out some key things. So these are actual frequencies we see in nature, and then these are expected in uniform, right? So just take these combinations and randomly sample them. Would it get some sort of curve, right? We'd be a lot flatter than what we see, right? So again, there's clumping. Okay, so far Stebbins looks like he's right. Okay. And here's the presumed root state for angiosperms, and it's still a pretty high frequency. Okay, so angiosperms started in that combination and continue that combination. Okay, so that's all good background, shows there might be something here. Let's go to the phylogeny. All right, so first we need a model, right? And so just take a binary trait model. So you have to think about state 0 and state 1. So you're familiar with like BISI type models, so starting off with that sort of approach. Right? We think, okay, what drives you here to here? Well, natural selection. Right? Natural selection drives everything. Well, it could also be drift or mutational bias. Right? Those things could also matter. Okay, and create things. So, for example, we see cavefish, they tend to lose their eyes. Right? Now, it could be natural selection, why they're producing this expensive thing. It could also be just drift, the genes that help you produce eyes, they go away. There's no selection against that. Okay, or mutational bias. Now, this affects frequency of 0 and 1, but also things can affect this is speciation and extinction. Right? So if that stuff in state 0 speciates more, I'll start seeing more and more of those. Or if stuff in 1 goes extinct more, I'll see fewer and fewer. Right? So the frequency of traits I see in the tree of life are based on all these processes happening at once. Okay? Not just for a single character. How about for combinations of characters, which is a real question? Well, we can extend this. Right? So now we have 0, 0, 0, 1, 1, 1, 1, 0. Okay? And again, we have rates going this way, and so forth. We assume we can't go here to here. You assume we can't change two characters at the same instant, right? You might have to go here on Monday and here on Wednesday, right? But still, there's a little tiny lag. Okay, this is controversial. Not everyone thinks it's something we'll want to do directly across. In some traits, that actually might make sense. Okay. So, there's all the factors that can lead to this. Well, no, there's also chance, right? We know that evolution is also stochastic, right? So you might see lots of 0, 0. Not because it's awesome, it has selection for it, although it's low extinction rate. It could just be by chance we started off life that way, with the plants that way. It hasn't had time to move away from it. Okay? So we can lump these into intraspecific processes and then interspecific processes, time, chance, things like that. So 2 binary traits. We have 8 transition rates, 4 birth rates, and 4 death rates. Okay? Well, we're looking at more traits than that, right? So we're looking at, say, 3 binary traits, 12 transitions, 8 birth, 8 death. 4 binary traits, 64, 16 and 16. We're looking at 16 binary traits, right? So we have 384 possible transition rates, 64 birth rates, 64 death rates. Estimating all of that would be stupid, right? Unless you have a giant tree, right? You're not going to have enough power to estimate all those things. So here's our dilemma. We care about the combinations. We can't estimate this full model. Okay, so what do we do as a hack? Okay. And our solution was focal areas. Okay, so for example, if I say, let me think that what matters is having a corolla present, having bilateral symmetry, and having few stamens. Okay, so looking like an orchid, right? Sort of structure, right? I think that's an awesome phenotype. I think that might cause higher speciation rate, lower extinction rate. Okay, if that's true, we can try looking at just those plants that have that combination versus other plants. Right, so just stuff that has those particular combinations there. Right, so we can convert this into a 2-state character again. Right, blue don't have that, red do have that combination. Okay, so that focal area. And so then this crazy, scary network becomes a slightly less scary network. Right, it has only a few colors. And I can separate it out, okay, where I have all the taxa that don't have that combination and everyone else, right? And they have transition rates going from one to the other, and transition rates within. Okay. And now my model is a lot simpler. Now I have a birth rate in my focal state, a death rate in my focal state, birth and death in non-focal, and then transitions within focal or between each. Okay. And then I can think of various ways to restrict these even further. So now I have, for this particular focal area, a much more workable model. Okay. But of course, there's many such focal areas, right? I could just say corolla present. Just look at that one, right? And then you have this arrangement. Or 4 characters like this, or a pair of characters. Okay, so we've lost some complexity in terms of the model. In terms of parameters, we've gained some complexity in the focal areas. So how bad is it? So 5 transition models by 6 diversification models by 3 to 6 focal areas. It's a big space. Okay, once you throw out the ones where we don't have enough data, we get down to only 19,000 models to look at. Okay, so here comes our carbon footprint. Right, so we fire up the cluster, start running things. Okay, and so what we did was estimate the likelihood using diversity in a particular model. Right, so we're a script to do this and loop over all those different models. Okay, and then calculate A, C, and Akike weight. Okay, and here are the results. And so it tells us which focal areas are important in the models. Okay. So corolla present, bilateral symmetry, and stamens few, like orchid. That models that have that and have this diversification parameters and transition parameters have a pretty high weight. Bilateral symmetry alone also matters, right? Or also, individual pairs of those. So bilateral symmetry, stamens few, corolla present. We're seeing this pattern, right, where these three traits seem to matter a lot, right? Whereas other traits were not selected much. Okay. And so we can plot just those. Okay. So here we have those traits, and you can see, so this triangle has all the characters that have corolla present, bilateral symmetry, right? And so, sorry, color is diversification rate, right? So there's a very high diversification rate. The medium ones nearby, as you lose bilateral symmetry, it gets slower. Okay? And then circle area is proportional to the amount of species in that state we have now. Okay? So even though this is faster than this, we still have a whole lot of these. Okay. So cool. So are we done? No. Right? First of all, you should worry about, did this happen once in the tree? Did it happen many times? Right? Because various issues come up with that. And so we can plot that on the tree and find out that combination of three characters has evolved multiple times. Okay? Does it mean we're safe? No. But it helps give us some confidence. Okay? So have we solved all this? Well, no, we looked at this and this and this. We've missed time and chance. Okay? Why should we worry about this? Well, if you look at the actual frequency of just looking at these three combinations, right, our equilibrium frequency, so we just took these rates, ran it forever, how many of these taxa would you see? It'd be this black bar. Okay? We see the gray bars. Right? Well, look at that mean. Look at mean our model is wrong, which of course it is. Right? I mean, we know life is much more complex than this. There's things like mass extinctions. There's things like growth habit. This model is a bad model. Right? It's a pretty good one. It's a complex one, but still doesn't match everything. Okay? So that could be one aspect of it. Another aspect, though, is that maybe we're not in equilibrium yet. It just takes a while to get there. Okay? So how can we deal with that? We can rerun the tape of life. The idea from Gould. So take our parameters and actually run a simulation simulating hundreds of thousands of species on a tree and see what we get for these frequencies. Okay, and so here we see, let's show you some annotations of this. These black marks show the observed frequencies. Okay, what we see right now. This is simulations up to the present. So each line here is an individual simulation. Okay. And these are the distributions from the simulations, right? Both now and 15 million years in the future, right? So why stop at the present? Why just keep running in the future? See what the future is going to be like, right? So you are here, right? We go into the future a little bit. Okay. And so we ran this to sort of see how these things are changing over time. Okay. And what we found was that the root combination, right, even though it has a lower diversification rate, it's a long time to disappear. Right? Even though the other ones are cranking, this is the state we thought was the optimal state in terms of maximum diversification rate. It takes a long time for it to appear, and then once it appears, it takes a long time for it to become fixed. Right? So the reason we're not at the equilibrium yet is because we're not at equilibrium according to this model, which is kind of cool. Right? Whenever you find these models, they sort of assume equilibrium. I mean, think, 130 million years is a long time, but it's still going to take even longer for the traits to stabilize. And one reason for that is it takes a while for them to appear. So here's these trait combinations. Here's the assumed ancestral combination, right? You can see how long until these different combinations appear over evolutionary time, right? And it can take, tens of dozens of years, millions of years to get to this optimal combination. So one reason you don't see it is because it took so long for it to first appear, we think. Okay. Now this generates a prediction you can then test with fossil records and things like that, which we haven't done yet. Okay, it's testable. Also tell us something about how good our model is, right? So we have, from the equilibrium, here's our observed, and we can see if our simulations include the observed, right? And not really, but not horribly bad, right? That's been saying, yes, we won, right? But it's okay. Like that bar includes that bar and stuff. So it still shows we still have more to do, right? But we'd be on the right track. So summary of this part. So bilateral symmetry is important for floral diversification, but so is having few stamens and having corolla, right? So the character combination matters, right? Why might that be? It could be precision of pollinator pollen placement, right? And that could possibly lead to faster speciation rates because I put my pollen right here on this species and right here on some other species of pollinator. And so when I go to a new flower, it doesn't pick up pollen in the right place. It can lead to this reproductive isolation. Okay, so that's a cool testable idea. And then also different diversification rate, species sorting might be more important than transition rates. It's not something I emphasized here, but it certainly seems that what's leading to this different diversity is different speciation and extinction rates rather than biased transitions into a certain character combination. Okay, so cool. But caveats, small data set size, right? I mean, so for this thing, we had skilled botanists go and we say, okay, find this species and find out what its traits are. So go and look in herbaria or look in plant books, and so it took a long time. Even so, it's 500 species, not a lot for this sort of study. Okay. The Madison Fitzjohn points, right? We sort of hand wave and say, look, it originated multiple times, so we're okay. We're not okay, right? It could be that one clade is driving it, and there's some tests you can use to get at that. For example, you can split the tree up into separate independent groups and try each one. We haven't done that yet. Okay. That's something to worry about. And finally, there are possible, well, actually certain things we're not including in our model, right? So growth habit, being herbaceous versus being woody, that matters, not in our model, right? Biogeography matters. Rocks from outer space matter, right? None of these are in our model, right? So these are approximations that should make you a little wary of the results, okay? Even so, sort of promising. All right, so that's subject one. So if you want more, read the paper. I really love this paper. It's a good group. Okay, this is okay too. Now, this is the next part. So we understand phylogeographic processes. And this is work with Brian Karstens, me, Ariana Morales, a grad student, and Nathan Jackson. So here's the question. So I come and I have this map of a landscape, right? Who's reading the names? They see chuckles. And I have a group of specimens on this landscape. We actually use one of these in a grant, actually. And I have these specimens on there. I want to figure out what's the phylogeographic structure here, right? Do I have, this mountain range, the Misty Mountains, are they separating gene flow here, right? Do I have the Indian River? Is that causing a phylogeographic break, right? So I'm coming in without hypotheses, right? I want to say, what's going on here? How can I figure this out? Okay. So what could I do? Well, I could group them by populations first. Okay. Here's all one contiguous forest, one contiguous forest, something goes into discrete populations. Okay. And now I have relates to landscape. Now I have my samples and populations. Okay. And what comes naturally here? Well, making a tree, right? It's one thing that's awesome about University of Idaho. You guys know about trees and phylogenetics. It oozes from the place. It's good. So I can make a tree. So it might be this tree, right? It might be this tree. It might be this tree. Are we done? No, right? We also care about population sizes or phylogeography. Maybe we had, big population size here, and then something rose in the east, and population, became smaller, right? What else can happen? Well, it could have gene flow between populations, right? That might affect the phylogeography, okay? I might even have a tree structure, right? If, all the lineages here coalesce because of mutation, because of migration, rather than due to a shared history, right? So the fact that they collapsed at some point in the past doesn't matter. Sort of wash that signal out. So all I need is an island model, right? And that's the extreme here, right? So you can imagine all these different models you could look at. Okay. And where else have you seen this sort of thing? Well, maybe in sort of model test, right? So those of you who are, you don't do this that much anymore, but if you're looking at phylogenetic models, you can say, let me churn through a whole bunch of models. This model test will tell me which model is right. Okay. And we see this sort of thing in a lot of areas, right? So we have, ideas we have specified hypotheses, like I want to test this model versus this model, right? You can do that in PAML, you can do that in OUCH, you can do that in LASER, you can do various ABC approaches in phylogeography, right? But I want to come in and say, data, tell me what hypotheses I should have, right? Tell me what models you like, right? Those models, this program's that, like model test, or code test, or surface, or IDA, or producer, or BAM, right? And then phylogeography, now we have FRAPL, is our thing. Okay. And so to solve that use case, right, you have this question of what could be my phylogeography, help me figure that out, right? And one cool thing about this in general is that you can be surprised. Right? Oftentimes what you do in phylogeography is say, here's my model I love, and here's a stupid null. Are you better than stupid null? Yes, you are. Yay, I know something, right? We're never surprised, right? Whereas if we do this, you can go out and test a whole bunch of models and find, wow, that river is not a barrier. That's really cool, right? Now it could be you're wrong, right? At least you're learning something, and you can then test that more and more with other data sets. Okay, we need more surprise in biology. So Frapple, phylogeographic inference using approximated likelihoods. So we worked hard at this. NASA quality acronym. The input for this are gene topologies, or SNPs, which are sort of like really bad gene topologies, how we deal with, and an assignment of samples to populations. We say all three of these came from the same area. Some of the jargon we're going to be using: N is the number of populations, and then K is the number of free parameters, so sort of AICK. Okay. So what do we do? First, we generate all possible models. Okay. In this large number of models, we have also now approximations. We can do fewer. Right? But the idea is you generate all these different models. Really, all these little cool renderings are made in R using the program. So this is sort of built in. You can actually print them out. I have a printed out version at home. You then filter them. Right? So you say, only show me tree models. Only show me models that have no more than one migration parameter. Okay? To make it sort of feasible for your data set size. And then you analyze. So you find the best model, you can find the AIC weights for each one, right, and compare that way. So what does this entail, generating all possible models? So think about just three populations, right? What could happen in those? Well, I could have, all of them coalescing at the same point in time, right? I could have A and B coalescing, B and C, A and C, or I could have none of them coalescing at that point, right? And what could happen next? Well, I could have, them coalescing this way, or this way, or this way, right? So you have these sorts of models, or not coalescing. Right, so for three populations, here are all my structures of these kinds of models. Okay? Now, I haven't added on things like population size or migration rate, or those sort of changes that also can matter. Okay? And so, looking at number of free parameters, here I have one free parameter. How deep is this split? Okay? Here I have one free parameter. How deep is this split? Here I have two, this split and this split. Okay? Just like this model. Now I have, for this branch, I say let's population size be N1. Okay? Now this branch could be the same or different from A. If it's the same, it's still only one parameter. If it's different, it's a second parameter. Okay? This one could be another parameter or not. And so forth and so forth. Okay, so you can see how this blows up. Okay? Once we have this model, right, then decide about migration. Okay? So I have this time interval here and this time interval here. Right, by three populations. So here, I've merged AB into AB. Okay, and now I have all these possible migration parameters at that time. Right, and again, I could have this migration set to be 0, or set to be a free parameter. Right, this one could be 0, or match that one, or be another free parameter, and et cetera, et cetera, et cetera. Right, and so in this case, I can create a model where I have just migration from C to B, and then reciprocal migration from AB to C. Okay, that's one possible model. Of course, what we do in reality is try a whole bunch of these models. Okay. So once you have that, you can then visualize that model and look at it. So you could just do a boring 2D plot, but why bother? Just go 3D right away. And so you can see so we have to show color for population size. So B is a different population size than everything else, right? And then you have these migration rates. So that's one particular Frapple model. And so number of models, so it's scary, right? Two populations, stabilize pretty easily. Population goes up, population goes up, 5, 6, 7, be careful. And so when you get to that point, you definitely need to do subsampling. Okay? And you might want to try in that case like a classic island model, a classic phylogenetic model, and then various models between. And just zooming in on what's happening here, populations, with this number of free parameters. You can see different models, different types, right? So we have number of parameters used for collapse. So we have two populations becoming one. The time of that is a parameter. N, population size. We can have 1, 2, 3, 4. And migration rates. How many migration rates do we have? Right? And so you can see, I can look at like this model right here, the simple model. We have no collapse, just migration, right? I can have this model here that has lots of migration parameters and a few collapse parameters, and one collapse parameter, and so forth. Okay? So each of these little bars is a model. So I can filter them in various ways, right, to make it a feasible working space, right? And then I can do my analysis. Okay? So how do I calculate the likelihood here? How do I calculate this? And so what we'd ideally do, what seems natural, is exact likelihood, right? So you just plug and chug. I have the probability of the gene tree, given the population history, calculate likelihood. And you can do that in things like coal, right? The problem with that is it doesn't always work. So it can be slow, and that's fine. You just be patient, right? But with some of these models, there's no closed form solution. So you can't plug and chug and get the likelihood. You have to figure out some other way of getting it. Okay. So you need to do that. So one thing you could do is something like approximate Bayesian computation, right? And so, sorry. So here's the basic algorithm for the ABC, right? So for many reps, you sample from a prior, you simulate using that prior. If some summary stat of your data set is less than some epsilon distance from your summary stat of your simulated data set, you count as a match, and you say the parameters, and then repeat. That's basically ABC. And of course, there's many cleverer ways of doing this. This is a dumb brute force way. This is a raw way of doing it. Okay. So problems with ABC for this, though. Model choice can be hard with ABCs. It can be good for parameter estimation, but choosing between models, it can have trouble. Okay. And the other thing is it's Bayesian has priors, right? So phylogeography matters a lot for conservation, right? So are all these small little species, one population is like one big species, and so I could put a Walmart here, or are they little species that need to treasure, right? And that matters, right, in terms of conservation priorities, understanding of how biology happens. Okay, and so by having priors, you can introduce this human element, right, which can be good in some hands and not good in other hands. And so if you can get rid of that, that can be an advantage for this. So we use approximate likelihood, right? So we estimate the probability of data coming to exact matches, and it's pretty similar to ABC. I'll walk through how it's different. Okay, so here's the ABC algorithm again. Now let's modify it. Okay, so now we sample from guesses rather than from priors. Okay, and we're also doing, rather than doing a summary stat, right, of, is this within some unit of it, looking for an exact match. So does my topology match exactly? So I simulate a gene tree. Does it match my observed gene tree exactly? Okay, we'll talk more about that in a minute. You should be getting worried about that. You should be scared. But you should, once you're done, you should feel a little less scared. But still, okay, and then count as match, and then we don't care about the parameters, right? We're actually using this to calculate likelihood. So likelihood is the probability the topology is observed, the probability of the observed topology given the parameters, right? Which we approximate by the number of matches with the number of simulations. Okay, so it's like if I didn't know how to do binomial and said, okay, is my coin fair? I can flip it many times and get an estimate according to models. Make a new coin that has probably heads of 0.5, 0.6, 0.7. Flip it many times. The often I get exactly the same distribution of heads as I get on my coin, right? So that's approximate likelihood. Okay, so that's cleaned up a bit. That's our algorithm. Okay, so just comparisons. So summary stat is a function of the data. We're using topologies, sort of is the data, or are the data. Now, arguably, you could say we're using a topology as a summary stat, right? So it's not worth getting into a religious war about this, but people do disagree about this. Doing simulation, approximate match, exact match-ish, priors and the starting values, we're doing grid search, and then integration, taking the maximum. Okay. So how we do this, we take our model, then we take our parameters, right? So collapse 1, how deep is the split? It's 4 coefficients in the past, which says here is 1, here is 3, migration rate is 0.2, migration rate is 0.6. Okay, so what do? You stick it into Dick Hudson's MS program, and you don't have to do this yourself. The program does it for you, but you want to give him credit, right? So input all these things, and then it simulates many trees, right? And then the program sees how often do I match my observed gene tree with one of those simulated trees. And that's the likelihood of that observed gene tree. That's it. Now, this seems like a dumb idea, right? So this is a log scale, right? So in a log scale, exponential growth, nice and linear. Right? Cool. The number of trees goes up double factorially. Right? That's very scary. Right? And so what's the chance of, when you have a moderate-sized data set, of having an exact match? given that given that each tree is as frequent as one of the atoms in the universe, right? Well, what helps us with this is tree probabilities aren't uniform, right? So not that they have equal chances of any topology. It's that if I have a given phylogenetic history, certain trees are much more likely, right? So if I have an example of a very nice phylogeographic history, we have nice, long, skinny branches, right? So that all the gene trees match the species tree, right? We're probably getting the observed tree is probably 1, right? Now if I have a short, very fat thing, then we're in trouble. Okay? So that's one area that helps us. Okay? And we also have other solutions. Okay? So I don't really care about labels within populations. I just care about relationship between populations. Okay? So what I can do is relabel those and then correct for that relabeling. Okay? So do that as well. Okay? Another thing is polytomies are soft in gene trees. Right? So if I have an input tree that has some polytomies, that could resolve to this tree, or could not resolve to this tree. Right? There's possible resolutions for this. And so I figure out how many of those possible resolutions will match the simulated tree. Okay. So that's how we can do SNPs, right? Because a gene tree from a SNP is basically a really bad tree with just one internal branch, right? And then you can figure out how that would match your observed gene tree. Okay. Another idea, which I was resistant to at first, but actually seems to work, is subsampling. Okay. So rather than taking every sample, you can sample at random and make the gene tree smaller and more feasible to work with. Okay. Now it has a trade-off, right? Because you have less power, but on the other hand, you can better estimate this likelihood. Okay. Another clever idea, you do many empirical loci, some are the same, right? So in terms of speed up, as you can go faster than linearly. So you can have really, really big data sets. It'll still run really long, but it won't be increasing factorially or exponentially. It just increases linearly. Or basically a little better than linear. So that's good, right? So for next gen stuff, you can use this. And then if you have other ideas, let me know. Right? I was looking for that. Okay, so analyze this. Okay, calculate likelihood using this. And now the question is, these are also parameters in the model too. We have to optimize those. So for a given phylogeographic history, we now have to estimate these parameters. Right? And there's ways to do this, and the thing we've come up with that seems to work well is a grid search. Right? So this is the dumb brute force way of looking at things. Right? So you have this unknown surface you're trying to find the maximum of. Right? So one thing you can do, most programs will do, is start with a value, try another value. Is it better or worse? Better? I take it. Right? Do it again. Better, worse? Not better. Better? Okay. And then finally, it stops. Okay? And there's various elaborations on that. You can look at like the gradient. You can have like little things spreading through, things mating and having genetic algorithms, and they crawl across and talk to each other, and all these beautiful things. And we did that first. That didn't really work well. Okay. The one thing that I think matters for this is really this approximate likelihood, right? So I look at this point and say, what's my likelihood? Simulate and figure it out. Okay. Now I try another point. At this point now, the likelihood is a little different, right? But again, it's estimate. So it's like trying to find the highest point in the ocean, right? You have all these little waves. And so the more you simulate, the calmer the seas are, it takes more time. Okay, to try to optimize across a possibly flat area that has waves can be hard. Okay, so what we do then is we just try these evenly distributed samples to find out which one's the best. Okay, here's my maximum likelihood. Now, one side benefit of this, though, is now I've sampled many points, and I can look at the support for these other models, like other parameter values, and say, okay, here's the best one. This is also still pretty good. So this gives me a sort of estimate of the uncertainty at the same time, okay, unlike a search. Now the downside is, it could be the true best is over here, right? And I haven't sampled that point, so I'm missing that. So you could imagine then, once you find out it's over here, then start sampling more intensely around, or that sort of thing. Okay, so there's ways to get around that, but it's something that requires user input at the moment. Okay. So how have we done this? So it's coded in a mixture of R and Perl and MS. Okay, so this whole kludgy thing. People in that row are now saying, okay, just do it in C++. Yes, you could. Go for it. How does it work? Okay, so here we have different models, right? So here we have isolation-only models. Here we have IM, so isolation migration, right? Here you have mixed models, right? And here we have migration-only models, okay? And black is when we pick the exact best model, okay? And so as we get more loci, we do a better job, right? For some models not that good at. That's worth being cautious about. Okay, and we're being very strict here about getting exactly the right model. So if you have a model that has, misses a small migration rate, and if we say the best model has no migration rate, we're saying we're wrong there, right? So try to figure out exactly how to measure how good you are. So we're being very strict about absolutely right or not. But that said, I mean, you should still be cautious, especially if you're using few loci. Okay. And here's an empirical example. So we have 3 bat species, right? So by Ariana Morales Garcia, and she looks at the weight of the best model. Okay, so we don't know what the true model is. This is real data, right? But we're trying to figure out what's happening with these bats. And so she tried more and more loci and saw how this converged on this complex model. We have ongoing gene flow between many of the species. Okay. And so it's cool because rather than saying, well, this is an island model only, or is the model we have just a branching structure, we found out there's branching plus gene flow that matter. Okay. Now, if you want to, you can go back and look at this with more detail on other methods. Okay. So summary for this part, it's a way to create many models, right? You can evaluate them, right? So you don't come in with your pre-existing beliefs. You can say, nature, tell me what's happening. Nature will say, maybe this. It's prior-free, but complementary to other approaches, right? So if you find out that the 3 best models are these 3, you take them to IMA or take them somewhere else and test them there. That's fine. Okay. Should scale pretty well, so as you get next-gen data sets, it should work okay. It can be used also for phylogeography, and we have a paper out now about species limitation. Too, if we want to figure out, whether it figures out, how many species there are of something, this can work for small numbers of taxa. Okay. And we tested against BPP, which is the gold standard. We found that, humans right now are 1 species, which we have enthusiastically shown, right? Where BPP says humans are different species, which seems a little off, right? Where it sort of tends to oversplit. There's another paper that came out recently that shows it tends to oversplit. Okay, caveats though. So on scales up to about 5 populations. If we want to do it on 200 taxa on a tree, you have to think about other ways to hack it to work that way. It's going to be hard. Okay, it's not using any information about branch lengths. Okay, something reviewers criticize because if a gene tree has this information, we just look at the topologies alone for the exact topology match. Okay, throwing away information should make you feel bad, right? In this case, you find it doesn't seem to help you much. But there are cases where it might actually be useful. We have some identifiability issues. So having a deep divergence with gene flow tends to give the same sort of shaped gene trees as a shallow divergence with no gene flow. So you have the program tell us apart. Okay, so that can matter. And in healing cases, there's 0 matches. So if I simulate 10,000 trees and none of them match my observed tree, the likelihood of my tree is 0. So log likelihood is negative infinity. It's hard to work with negative infinity, right? And so we have a kludge to fix it, and if your program sees too much of it, it gives you a warning like, hey, you need to simulate more trees, something's going wrong. Okay? But that could matter in some cases, and so it's worth worrying about that as well. Okay. Now, last part, better models of protein evolution. This is Mike Gilchrist, me, Jeremy Liu, and JJ Chai. Okay. And here's the central idea. So you're a cell. You make a certain amount of product per day. You want to make, 1 load of wood per day, right? So you need to have proteins to do this. I have to keep producing proteins to do this. Okay. And if I have an efficient protein, yay, gasoline, it can cut things down really fast, right, and produce at a very efficient rate. So there's a maximum rate for this ideal protein, right? If I have someone who's lovingly honed an axe, right, that can take a lot longer. Okay. And so I might need to produce more of these workers to the same final output of functionality. Okay. And so our basic idea is when you have less efficient workers, less efficient proteins, you need to produce more of them, and that's a cost. Okay. So the all proteins are efficient as possible. So all the proteins are less efficient, so we need some way to have the same net functionality. There's a calculable cost to this. We can get down to the mechanics and figure out how many more ATPs you need to produce a slower protein. Okay. And then overall, you can figure out the rate of new mutations. Since we're probably fixing these due to the cost that we can calculate, get the substitution rate. Okay, so the overall idea here is going to use PopGen to get a substitution rate for phylogenetics. Okay, and fit that. Let's build up to that. So we have a DNA mutation matrix. So GTR if you like that, HQA if you like that, unreversible if you like that. Okay, here is a random GTR matrix. Okay, where circle area is proportional to rate. Right, so here I happen to have a very fast rate of going from G to T, and the same rate of going from T to G in this model, and the slow rate going from A to G. Okay. You use random numbers. Real data doesn't look like that, right? And now I have that for individual DNA. Now I can also make that into a codon matrix. Right? So what's happening here? So now I have going from AAA to AAC is just the AC rate, right? And like our previous thing, we assume you can only change one thing at a time, right? So I can't go from AAA to TTT in the same instant. It just has to change one way and the other way. Okay, so sort of zooming in, I can see these different rates, right? So the AA to AAC rate, AA to ACG rate, 0. Okay. So from this GTR matrix, I get this complex-looking codon mutation matrix. Okay. See the ways in which I'm introducing new changes. Amino acid properties. Okay. If you've seen this and you forgot about it, it actually can be useful, right? So think about things like, if you ever looked at like your actual data, so when you're actually doing phylogenetic data sets, if you convert them to amino acids and just look at what's happening. Right? It's always good to get a sense of your data and play with it. And you'll see if you have a column that has some I's, it'll have V's and L's in it too. Right? If you have a column that has Q's, you'll never see N's in there. Why is that? Well, because these are all pretty similar. Right? These are moderate size, hydrophobic, sort of fatty. Right? So if you're in a membrane, you can substitute this for this. Your protein still is happily in the membrane. Right? So they put one of these in there, now it's taken out of the membrane, and your protein doesn't work as well, possibly. It doesn't work at all. Right? So to do selection, I can easily go between these, not between this and that. Okay? And so these amino acid distances matter. And so there's ways of calculating these distances based on properties. Okay? There's a whole field about which properties to use. So the classic one is Grantham, right, an old paper. And then people who are geeks in the field will say, oh no, Grantham's horrible, we must do something else. And you can. So we've written this, you can put in your own properties and deal with that. By default, what we're just doing is doing Euclidean distance of the Grantham properties, and they just are weighted, right? So I have composition, I take the difference composition, times the weighting factor, square that. Difference in polarity, times the weighting factor, square that. So from the square root. So just Euclidean distance. Okay. And the cool thing about that is then for any pair of amino acids, I can get the distance between them. And so, cysteine is weird, and so it's a high distance between everything else, right? You see, it's weird. Right? Whereas, isoleucine, valine, and leucine are pretty similar to each other. So they have, a low distance. Okay? And again, just based on these, very simple table of properties and those three weights gets me this rich matrix. Based on the biology. Another thing that matters is expression level. Right? And thinking about their overall cost, if I need to make one wood pile per day, so having 2 guys for that, it's not so much worse than having 1 guy. Right? But making 3 per day, cost becomes greater. Okay. Now the cool thing about this is that then expression level can affect your substitution matrix. It doesn't mean you can estimate it using your data. Okay, we'll talk about that in a sec. So we have probability of fixation. So if I have the new codon, what's the chance of it actually being selected and maybe reaching fixation in my population? Okay, and it varies depending on what the optimal amino acid is. If I want an alanine, right, I have a better chance of going there I have a better chance of picking other proteins that are nearby alanine and sort of in distance. The proteins are very different from it. Okay, they're very different from it. Whereas if leucine is the optimal one, then I have a higher chance of going from isoleucine. And so depending on which amino acid is optimal, I get a different probability of accepting these changes. Okay. And again, the only thing that we're using here to estimate all this complexity is gene expression level, population size, those distances between amino acids, and the cause of protein synthesis. Okay. So again, get this complex model from a few basic popgen parameters. Okay. And then you can do is convert these back to the codons that underlie them, right? So if I know that this is represented by CCA, CCG, CCC, I can figure out the rate of going from any of those to another codon that affects a different amino acid, codes of a different amino acid. Okay? And so I can go back to this codon fixation probability matrix too. Okay? So now I have my probability of mutations, my rate of mutations between things, my probability of accepting them when they are mutated. So multiply them together, boom. You get a substitution rate matrix, right? And this isn't, Halpern and Bruno have done this. We're not the first ones to do this, right? But the cool thing what we're doing is creating these initial matrices from much simpler popgen parameters. Right? So rather than estimating all these different probabilities of change, we're just doing it based on a few, how different are these amino acids. Okay? So here's the matrix if alanine is optimal. Right? So you want to be alanine, but you can be something else, with some cost. Here's with leucine being optimal. Okay? And of course, there are 20 amino acids. So you look at these matrices, right? Looking at it from above, I sort of stacked them all on top of each other. Right? You need 3D glasses for this stuff. Look at the side, you can see, for some things they're all pretty similar, right? So there's similar substitution rates, but for other codons, they're very different substitution rates. Okay, so now I have this really, really complex model, but again, the very cool thing about this, so this really complex structure, right? It just comes from a few parameters I can estimate, right? 3 weights on amino acid distance properties, right? Expression level for the gene, population size, mutation rates, it's like a GTR model or HKY or whatever, and then cost of protein synthesis. Okay? And just from those, I get all these different matrices. Which matrix do I use at a single site? Well, it's based on which amino acid is optimal at that site. So I have to estimate that as well. Okay, so I say, at this site in my protein, the optimal amino acid is A. Not sure if it's V. Let's try if it's I. Calculate the likelihood of each of those. Okay. And eventually branch lengths and the topology. Okay, so cool. So we have this complex model. How does it work? Does it work okay? And so here we compare, on a yeast data set from Stichels and Rokas, we compare GTR plus gamma, which we all love and use all the time, right? That's the model you use. It has, the 5 parameters. That's all you need for, phylogenetics. Well, 6 parameters you need for phylogenetics, right? Goldman and Yang, mute cell, and then R2 models, right? So SLAC, and then SLAC plus gamma, right? Which adds a whole extra parameter, right? And so we have the number of parameters. We have a few popgen parameters we're estimating, which is cool because you can actually get at those. We also have these optimal amino acids. And so what's generally driving this number of parameters is at each site, which is the optimal amino acid. Okay, and then we do what comes naturally: AIC, delta AIC, model weight. I'm like, yay, we win! Delta AIC of 395,000. That's bigger than 2 or 4. It's probably okay, right? So you don't talk about significance, but it's just sort of whoppingly better. Okay. Well, that's fine. We have a complex model that fits well. Does it actually feel like a real data set? Okay. And so what we also care about is model adequacy. Okay. So what we've done for a test here is take our tree, drop off a taxon, estimate all the parameters, estimate what the states would be at this point here, and then simulate through time using each of these models. And if the models are working well, I don't know, simulating, my simulated data set should sort of look like my real data set. It's sort of like a cross-validation approach. And so here looking at measure of functionality, right, with simulating different models. Okay, so I drop off my taxa, make up taxon, make up the data. So here's the truth, that dotted line, right? Using us, you see it come up here, and we're off a little bit, we're still pretty close. Other things have this decline of functionality. Same thing here, we're off a little bit, and we're diverging even further, right? These other ones are much worse. So are we hitting it exactly? No. So again, more work needs to be done, right? But still, it sort of feels like real data. We do other things too, other measures of how similar are the data sets. We also seem to win in those. Right? So again, both in model fit and also model adequacy, seems to be a pretty good model. Okay. So summary of this. So we can create complex models for a few about interesting parameters. Okay, so one actually fun thing about this is you actually infer, since expression level is part of the model, you can actually get a likely estimate of what expression level is for different genes. It actually compares pretty favorably. This is work by Cedric Lander, a grad student at UT. It compares pretty well to models that use like experimental evolution and seeing how genes evolve with antibiotics. Okay, you actually get a better estimate from using SLAC than from using actual empirical data, which feels deeply wrong, but that's what the data are suggesting. Kind of cool. Outperforms the models we typically use, and also links popgen with phylogenetics, right? Because they're all DNA evolving on the tree is evolving in a popgen model. Let's put that into the tree. Okay, some caveats though. Assume suboptimal proteins can be compensated for by producing more of them, right? So I have a guy with an axe rather than a chainsaw. I just need more guys with axes, right? A guy with a coffee cup, right? He's going to be wailing at that tree a long time and not getting anything, right? And right now our model assumes you just need 3 guys then, basically. That's a problem, right? So this probably works best when you're sort of near the optimal near the optimal protein. you get something that's really bad, and all probably fits really poorly. I think we haven't checked yet, but sort of feels that way. Okay. It's a slow implementation, like really slow. Like we need a tree for 5 taxa, yay, right? And part of our grant for this is actually have a hackathon and have people who actually know how to code put this into RacksML and Beast and things like that. Okay. Does not yet deal with code usage bias, linkage, and all sort of cool pop gen things that happen, right? Those things we're going to be adding. Try this as the same optimal amino acid for all taxa for all time, right? And I don't know, you often use that approximation, but actually you can start doing cool biology if you break that assumption. Say, okay, let's see, deep sea organisms. Do they have different optimal amino acids for their opsons? Let's see. That's one thing we're going to be doing with the hidden Markov model. Okay. So again, those are all the topics, and if we where to read more about them, or download the source code. So thank you. Yeah.
So when you say it assumes the same amino acid is optimal for all time, it's basically ignoring epistasis entirely.
Yep.
Within protein epistasis, between protein epistasis.
Yep.
And it's also ignoring change of function.
Yep. It's not just ignoring it, it's assuming that it's 0. So it's making a worse assumption, right?
And so
I like to oversell stuff.
Good for, I'm trying to figure out when you could use it.
Well, because right now what we'd use is a GTR gamma model. So a model that just assumes we have, the basic, that 4x4 matrix, and that explains all of evolution. And so this is richer than that.
So this is like, I want to use DNA data.
For protein coding genes.
But I want to take into account silent mutations, not yet codon bias. But
Right.
Yeah, okay, I get it.
Yeah.
I got it.
Good. Do you like it?
I do like it for that.
Okay.
I wish you'd work on the other parts.
Well, we're working on it.
I know.
Other questions? Yeah.
I'm curious just about the biological interpretation for the floral combination of traits stuff, the first part.
Yeah.
And so is that analysis suggesting that all flowers will end up with those 3 traits?
Not all flowers, but like that's like the equilibrium frequency is not 1, but it's pretty high, right? Now, of course, you could imagine that will that doesn't work. Like some environments, it's probably going to have wind pollination and stuff like that, and so it shows the simplicity of the model. But would you buy that we're not there yet? That things are moving in that direction? Like when it comes back.
Certainly, if you look at the distribution of those traits, those are the most diverse things. I guess what I'm thinking is that there are a lot of other underlying parts that could be influencing diversification rate to give you lots of species with those particular traits. So that combination of traits itself might not have very much to do with the diversity that we see in those clades.
Right, and this could be the Madison and Fitzjohn thing again, right?
Exactly.
So it could be that, like, yes, we see this occurring many times, but what's driving it is orchids are awesome because they smell good or something. That's why they're not going extinct.
Yeah, or all of these are paleopolyploids or something like that, and that's really what's going on, and they just happen to have that combination of traits because the ancestor of that had the combination of traits.
Right, and so it's hard to. So this analysis doesn't get at that. Now you could imagine analyses that do. So I have a student in the lab working on parasitic plant evolution with Chuck Davis, and we're looking at splitting up a tree and looking at multiple repeated instances in seed. You see the same pattern every time. So we're getting back at the number of changes. So yeah, they won't work well here because you already have only 500 taxa. So splitting up further, if you find something wrong, I probably won't believe it. But, yeah.
Does the order of which traits evolve matter?
So this model assumes that the rate itself does not depend on the history of the trait, right? But what we see today depends on what evolved first, right? So the transition rates are very low. So it takes a long time to get this combination to appear. Once it appears, it goes, yay, we take off, right? We have to wait tens of millions of years for it to come out in the first place. Okay, which is kind of...
There's no rates for the... Did you look at any of the double combinations before getting to the triple?
So the cool thing about this set of models, we look at the singles, we look at the doubles, we look at the triples, we look at all of them, and it found out that the triple was what mattered. Also, but the next one down was some of the doubles. Yeah.
So when you factor, so you've shown like the best models are far away from the equilibrium state, but what if you encounter uncertainty and you like reject the rapid equilibrium?
So it's like, what do you mean, like the uncertainty affecting the equilibrium?
Well, so there's uncertainty around those models, right?
Yep.
Parameters, and you maybe reject the assuming you're using one set of parameters to do those solutions.
Well, we're using the model average one, but yeah, still one set. Yeah.
Right. So does the if you were to put a 95 element set on the set of models and parameters, would you still reject?
Yeah, I haven't done that test, but almost surely you would. Yeah. And I think it makes sense that life isn't at equilibrium. I mean, but it's not something we often think about. There's an advantage of having this group where we're sort of chewing over these ideas among people of very different backgrounds for like 8 years. So like look at all these different aspects and sort of get at the biology. It's kind of cool. Bad way to get tenure, but still fun. Good biology. Other questions? Yeah.
So you said a couple of times that you've been basing your models on the idea that only one thing changes at a time.
Mhm.
I remember specifically from the protein evolution because it's the thing we talked about most recently.
Yeah.
You were saying that like only one one codon position can change at a time.
Yep.
Were you basing that on is there specific evidence for the fact that that's the way it happens in nature, or was it just because that's the only way to generate a workable model?
I mean,
It's
there's
not ridiculous and complicated.
many mutations do work on a single base at a time, but there are mutations that work on multiple ones, right? So it is an oversimplification. Yeah, but it keeps it simpler. Now it's less justifiable in some cases with like the other kinds of data, right? So you can imagine that you could have some mutation that changes the flower shape radically and affects even one Hox gene affects not Hox gene, but equivalent affects multiple things at once. Then that could cause an instant change, and our models ignore that, right? If you really believe in that, what you could do is put that into the model and say when we have a transition rate between these directly and compare that to a model where we have 0, which is better. Yeah. Did you want to?
No, yeah, that was where I was going to go. In that first example, changing 2 of those floral traits at the same time, there is definitely argument about whether the way they did it, with just having one at a time versus including those diagonals, is worthwhile. Part of it is a model complexity issue. It really blows up fast every time you have all those diagonals.
Yeah.
But you could imagine there are floral development genes that control both sepals and sepals and petals and stamens, and so you hit one of those, you might change all those characters out.
Yeah. Another thing caveat I failed to mention was like just discretizing itself is hard. You go in nature and you see, I mean, any trait that's changing that's changing nature changes within the species, right? So we only have some species that are variable in the trait.
Yeah.
Plus, you have any trait that's going to be hard to say, are you inferior or superior ovary? Well, how am I? And so there's always that fuzziness.
Right.
Yeah. Other questions? Yeah.
For the protein evolution model, I'm just thinking, when is going to be for real life, lots of data. So
Whenever you start coding it.
how well behaved is this actually on surface, and is that lots of correlation between these parameters?
So we've been playing a bit. I mean, one reason it took so long to get this out, we've been working on it for a few years, is like worrying about the likelihood surface optimization. I think our optimization is okay now, using NLopter and multiple starts and stuff like that. I haven't plotted the surface of the sea empirically how rugged it is. Yeah, I mean, there's no weirdnesses. I mean, we use log transformed weights and stuff like that, so there's no weirdnesses with like weird boundaries. Like you cannot get below 0 without flam, and that could cause problems with optimization. That said, though, optimization is always something I worry about. So, yeah. Does that answer your question?
All right, let's thank Brian again. And now it's spring break.